Hello, everyone. My name is Dr. Ashok Agarwal from Cleveland Clinic Foundation in Cleveland, Ohio, in the United States. And I welcome you to my talk on sperm DNA damage and assisted reproductive technology. So let's go through it. The main things that I will be speaking today are the limitations of current semen analysis. We'll be talking about need for sperm function test, sperm DNA fragmentation, how that has become such an important part of male infertility diagnostics, and its relationship with assisted reproductive technology as well as natural conception, and then provide you information on latest clinical practice guidelines for the testing of sperm DNA fragmentation in patients with male infertility. Let's begin now. So, what is the need for advanced sperm function test? The need is because the semen analysis, which is one of the first tests that is required on any couples with a male patient to evaluate their fertility, is a very poor test of predicting the fertility of that individual. The test has numerous problems. It is a very subjective test. It provides, in many cases, highly inaccurate results just because it is all done manually, and it's very difficult to get the results for many of the parameters of the sperm correctly, and the results are not comparable between different laboratories or different individuals who are conducting this test. So with that, you can see the results which we have reported in a publication. Here we are showing the overlap of the cutoff values or the reference values for main parameters of the semen, such as concentration, motility, and morphology. And you can see the lower and higher range of these cutoff values. And on the right side, we have the percentage of overlap that we see within the population of proven fertile men and patients who have male factor infertility. And you can see there is a humongous amount of overlap in the lower range, which are abnormal, compared to those which are higher, which are in the normal range. The same thing goes for proven fertile. They can have as many as 34% of patients which may be abnormal. If you look at the WHO cutoff value of 27.5%, at least so many numbers of patients can be having, and the same thing goes for motility, morphology. There is a lot of overlap between the two population of fertile and infertile men. So the semen analysis cannot really predict the fertility of an individual. And the same thing goes for the sperm morphology. So why it is? Because there are so many functions that the spermatozoa is engaged in after it is produced by the testis and released from the testis to the ejaculatory duct, and then through the process of conception transferred to the female reproductive tract. And you can see there are a number of changes that the sperm must go in order for them to be prepared for the process of fertilization. And these events are listed here, as you can see, which are necessary, whether it is looking at the membrane integrity of the spermatozoa or chromatin condensation, or whether it is morphology, whether it is capacitation, acrosome reaction, etc., etc., as you can see. So all of those events are very well synchronized. And if they are not occurring in the correct sequence, then those spermatozoa will not be able to fertilize and achieve further development. And that is a very important part, which is except for count, motility, and morphology, all other parameters require functional test because they are not the test that can be done as part of a routine semen examination. So if we look at the basic test The basic test is semen analysis, which are consisting of macroscopic and microscopic examination of human semen. But if you look at if the basic semen analysis does not tell us enough information about what is wrong with that man, then we have to go to the advanced test. So this is a decision that each physician has to make after they have done a proper examination and conducted a semen analysis. As I said, semen analysis is, even under the best circumstances, is a very poor test of predicting the fertility. It can only tell whether the sperm are present or absent and the spermatogenesis is happening or not. So looking at the advanced test, you can see a whole line of tests on the right side, whether they are sperm DNA fragmentation by variety of techniques of measurement of reactive oxygen species or mitochondrial function, or they are sperm zone pellucida binding. So there are a variety of things that are part of the functional test that can be done. Now, if you look at this particular figure, you can see on the left side the tests that were very popular before ICSI, which came into development in 1992. And many of those tests, which were very popular, are no longer conducted routinely. So whether it is acrosine test, acrosome reaction, which was a very popular test, or test for hypoosmotic swelling, or test for hamster sperm penetration assay, all of these tests have kind of disappeared. However, now with the development of ICSI, a new generation of tests have come out. They are sperm DNA fragmentation testing, the testing for hyaluronidase binding, or fluorescence in situ hybridization or fish assay, measurement of free radicals or reactive oxygen species or antioxidants. So these are new lines of test which have become more common now. And one of the most important tests is the test called sperm DNA fragmentation or SDF. And the test can be conducted by multiple different technologies, whether it is tunnel SCSA, which is called sperm chromatin structure assay, or COMET, or sperm chromatin decondensation. But this is not all. There are many other techniques that are there which can allow you to measure the number of sperm which have undergone any kind of fragmentation. So you can use fluorescence microscopy, which takes only 200 500 sperm are scored. It's subjective, it's time consuming. Or we can use flow cytometry, for example, for tunnel and SCSA. And that counts 10,000 sperm. And it is highly objective, it is fast, robust, and very sensitive. So when you look at the data from the literature, we see that SDF testing by tunnel is the most widely published technique in the world. And that is very important. So the types of DNA damage that we measure, which can be measured, are mitochondrial DNA damage, telomere attrition, sperm DNA fragmentation, Y chromosome microdeletion, and epigenetic abnormalities. So the most common defect is sperm DNA fragmentation, and there are techniques and methods that have been developed for routine diagnostic use. And this also represents the maximum or the major type of damage to the DNA. All other type of damage, except for Y chromosome microdeletion, is fairly small, and there is less amount of information on their routine testing. So we are going to focus our attention in this talk on sperm DNA fragmentation. The mechanism of sperm DNA fragmentation includes the following items, such as defective chromatin condensation, which is because of abnormal histone and protamine ratio, and that is the defective DNA repair mechanism, and that causes double strand break. Both strands of the DNA are having damage. If you look at the abortive apoptotic process, where the sperm with DNA damage may escape the Sertoli cell screening system, so they are not discarded, and these sperm also have double strand break. Now, oxidative stress, which is an imbalance of oxidant and reductant, forms 8-hydroxy guanosine and 8-hydroxy D-guanosine, and that results in either single strand break or a double strand break. So oxidative stress is one of the causes which can result in either double or single strand. So, in terms of whether the DNA can be repaired automatically by the cell, yeah, it is possible. DNA is repaired until the third week of spermatogenesis. However, after that, there is no repair capacity during its transit into the female reproductive tract and storage. In vitro experiments show that DNA damage does not impact fertilization, but it affects embryo development. And the human oocyte does have a limited capacity to repair sperm DNA fragmentation. Um, and through some lab experiments using radiation, we have been able to identify that up to 8% of DNA damage can be repaired in vitro by oocytes. So, at least in the laboratory model, using mice oocyte, and which have been where the sperm are radiated or exposed to 5 giga of radiation doses, that those sperm can be repaired in terms of the DNA repair capacity of the oocyte. But the oocyte capacity is declining worldwide. So there is really, I think, a lot of attention on sperm DNA fragmentation, and they can be measured by both direct and indirect tests. And these tests are a creatine orange, sperm chromatin structure assay, sperm chromatin decondensation test, tunnel assay, and comet assay. And they measure both single and double strand DNA damage. DNA damage can be evaluated by both microscopy or flow cytometry. So let's focus on tunnel. Why we are focusing on tunnel, as I showed in the first slide, in the beginning slide, that tunnel is the most commonly studied technique reported in scientific literature. It has the highest number of clinical and research studies. The principle of this test, it uses terminal deoxynucleotide transferase enzyme, which is a NIC and labeling. It quantifies the enzymatic incorporation of this enzyme into DNA. And the method is it labels nucleotides are added to the site of DNA strand. And then fluorescence is measured by a flow cytometry device. And as you can see, this is um, the normal levels of DNA, as you can see in the control population, and where we have been able to develop the cutoff value of 16.8 by using two different population, one a control, fertile control, and one a patient. So we have, and each center has to develop its own cutoff value based on the population of normal subjects and the patients that they have. So there is a poor correlation. People ask, why do we need, if we can predict from the semen parameters, sperm DNA fragmentation? But the semen parameters does not allow us to predict the amount of sperm DNA fragmentation in the semen. Uh, there is a weak relationship. uh, we know that infertile men have significantly higher values of sperm DNA fragmentation, but there is a weak relationship between the sperm DNA fragmentation and semen parameter. So, because it is an advanced test, which is independent of conventional sperm parameter, as you can see in this table, the correlation is rather very weak for semen parameters, concentration, motility, and morphology. And either the SCSA, tunnel, or comet, all three major techniques used. This is a meta-analysis to see the impact of sperm DNA fragmentation on natural pregnancy. And what you will see, meta-analysis is like a super analysis of large number of studies. It has over 600 couples who have high sperm DNA fragmentation, which were measured by SCSA and associated with failure to achieve natural pregnancy. So the odds ratio is very high, 7.01. What it means is that, and these are three large studies that were conducted, and you can see the odds ratio in all of them. Anything above one or two is considered pretty good. So here we have That means that there is, if the sperm DNA fragmentation is high, then the patients will have great difficulty in achieving a natural pregnancy. And a odds ratio of anything above 2 is very, very significant. And here we have more data. Here we have a meta-analysis uh, published in 2019, which included 13 studies with over 500 men with a history of recurrent pregnancy loss. Couples with a history of idiopathic recurrent pregnancy loss have higher levels of sperm DNA fragmentation than fertile couples. As you can see, this is a forest chart, forest plot, and these black diamonds are on the right side, indicating that the results are highly significant. If the red dots are in the middle, then it will mean that the results are not very conclusive. If they are on the left side, that means there is a poor relationship. But all of them are on the right side, which indicates that the results are highly significant. That means those patients who have high degree of sperm DNA fragmentation also have great difficulty, have a very high incidence of recurrent pregnancy loss. And then we studied on the impact of sperm DNA fragmentation on intrauterine insemination outcome and pregnancy rate. And this is a meta-analysis which looked at 10 studies and almost 3,000 IUI cycles. And the main findings here is that the high sperm DNA fragmentation is associated with lower pregnancy rate. So again, the results are very significant with a risk ratio of 0.34 for low pregnancy rate, and for delivery rate, the risk ratio of 0.14. Both of these values, as you can see, are highly significant. And you can see here that the results are being plotted on the forest plot. Let's go to the next study, which looks at the impact of sperm DNA fragmentation on IUI outcome in the pregnancy rate. Here we have three studies which looked at the high sperm DNA fragmentation, uh, and the number of studies in each meta-analysis is in this row, and then the number of cycles that patients underwent IUI are listed here. So altogether, almost 8,000 patients underwent this, and the main findings for each of them show that the odds of having pregnancy is reduced by almost 30% in patients with high degree of sperm DNA fragmentation by tunnel. And then in the second one, sperm DNA fragment damage is associated with low IVF rates. And then in the third one, comparing the effect of high and low sperm DNA damage on pregnancy after IVF, reduced pregnancy rate observed in patient with high DNA damage. And that last one, which was published by Simon and had almost 4,000 cycles of IVF, showed negative effect of sperm DNA damage on clinical pregnancy after odds after IVF with the odds ratio of 1.92. All of these are significant. Anything above 1 odds ratio is significant. So these results are very convincing. And then if we look at more studies, this is looking at the miscarriage rate after IVF and the live birth rate on the right side. So we have miscarriage and the live birth. So again, there is a very good correlation of high sperm DNA fragmentation with greater risk of miscarriage in three meta-analyses. The odds ratio range from 2.17 to 2.48, which is highly significant. If you look at the live birth rate, again, there's a significantly lower rate of live birth in men with high sperm DNA fragmentation following IVF with a risk ratio of 1.27 and a p-value which is significant. Again, I think let's look at the ICSI and sperm DNA fragmentation. Here we have the miscarriage rate, which on the left side, the miscarriage rate, high sperm DNA fragmentation of more than 30% by sperm chromatin decondensation test, one of the technique, is related with greater risk of miscarriage in ICSI, almost 40% versus 17. And that neutral comet can predict recurrent miscarriage with great accuracy. So again, these are very good results. In look at the live birth with the meta-analysis of 5 studies and almost 450 cycles, men with low sperm DNA fragmentation have significantly higher live birth rate or risk ratio 1.1. So conversely, men with high sperm DNA fragmentation have significantly low live birth. So these results are kind of matching what we have seen in other ART outcome studies. So how to interpret all these different findings? We can say that high sperm DNA fragmentation is related with decreased pregnancy rate, low pregnancy after IVF, and with increased miscarriage after IVF and ICSI. In ICSI, what is happening is that we are bypassing the natural selection process or natural fertilization. So high quality oocyte can offset the negative impact of sperm DNA fragmentation on pregnancy. That's why we don't see very good relationship with pregnancy in ICSI because of bypassing the natural fertilization. Also, gametes in ICSI are not cultured for very long period of time, unlike in IVF. And then there is a less exposure to oxidative stress condition, which are induced by laboratory manipulation. And the DNA damage may affect the genes responsible for embryo development and implantation. So we conducted a study and published this recently to document the clinical guidelines for the clinicians, when to and how to use the sperm DNA fragmentation. We have 24 international experts in male infertility who are involved in this particular consensus document, which was published in a very high impact journal. And it's a evidence-based recommendation for the testing of sperm DNA fragmentation. What are the guidelines? Here we can see these are the different clinical scenario where we should be using this particular test, whether it's recurrent miscarriage, whether it is lifestyle risk factors such as recurrent ejaculation, antioxidant therapy, lifestyle modification, or varicocele after varicocelectomy or in ICSI, recurrent IUI or IVF failure, also in unexplained idiopathic infertility patients. So these are the guidelines. Now, in terms of the take-home message of this lecture today, I want to say that sperm DNA fragmentation is an important functional marker of sperm chromatin, which really cannot be evaluated by a routine semen analysis, because none of the parameters of a semen analysis, such as count, motility, or morphology, can predict this particular marker. It's a molecular marker. It is not a marker that you can see under the microscope. It requires special type of testing, a special protocol before the results can be evaluated. SCSA, COMET, SCD, tunnel are some of the common tests for evaluating sperm DNA fragmentation. There is no consensus as to which test should be done by the clinicians or by the IVF lab. Each laboratory must make their own decision based on the scientific evidence that is around them, because the professional societies such as ASHRAE, ASRM, or other organizations have not provided any guidance. Even WHO has not provided any clarity as to which test is the most reliable test that can be used to evaluate sperm DNA fragmentation. High levels of DNA damage is associated with longer time to achieve a natural pregnancy and has higher miscarriage risk. And this has been shown by multiple studies that I had shared with you, whether it is with natural pregnancy, whether it is with condition of IUI, IVF, or ICSI. High sperm DNA fragmentation is related with decreased pregnancy rate with IUI and IVF, and with increased miscarriage following IVF and ICSI. This is very important to know that in case of ICSI, we are not seeing an impact on the pregnancy, but what we see is an increased miscarriage. As I said earlier, that in ICSI, we have a natural, we bypass the natural process of fertilization. So it confers some advantage, and there is less of problem with the pregnancy, but the miscarriage rate is very high. So sperm DNA fragmentation testing is most beneficial in the patients who have unexplained and idiopathic infertility, who are diagnosed with recurrent pregnancy loss, who have a condition of clinical varicocele grade 2, grade 3, who are opting for assisted reproductive technology procedures, and in those with lifestyle conditions such as smokers or patients who are exposed to certain kind of conditions during their occupation, who are obese, who have excessive cell phone use, or environmental risk factor where they are exposed to the variety of pollutants. So these are the risk factors where we feel sperm DNA fragmentation testing will allow us to discover hidden defects that are present in the spermatozoa, which are impacting on the results of ART and decreasing or failing the outcome of ART, and those are the most important conditions where this test will be highly beneficial. So again, I want to thank all of you for listening to me. This is our group at Cleveland Clinic, American Center for Reproductive Medicine in Cleveland, Ohio. You are all welcome to come and visit us next year once we are under the control the pandemic. So thank you very much, and all the best. Bye bye.